Good morning, everyone. I'd like to thank all the chairs and the admin for putting together this splendid event and inviting me. I'm having a great time. I wrestled with RDF all of yesterday morning, and I'm really looking forward to hearing from all of you over the next couple of days. I um, I thought I would bring you the news from a neighboring kingdom this morning. As Rutger was telling you, I come from the biodiversity data community. And that really is a big, broad umbrella. It covers a multitude of bins. No organism is deliberately excluded, although some are better represented than others. The research perspectives embrace everything from fisheries and agriculture through health, conservation, ecological modeling, and lots of evolutionary biology. We're experiencing a data deluge right now, and that will be important for a few reasons later. Um, it's not unusual. Most fields are, at the moment, flooded with data. The particular sources of our flood are threefold. The first is the past 200 to 300 years of natural history collection effort. It's hard to estimate how much material is in the hands of global museum community, but 73 of the largest institutions got together recently and tried. And per the estimate they published last year, it's something like a little over 1 billion items collectively worldwide. Each item contains some stuff like this, usually a physical object, a specimen or piece of an organism, and a bunch of bits of paper. How fun! There were efforts to put some of this information in databases in the 20th century, but the rate of maturation of tools and techniques means that nearly all the information currently digitally available was captured in the last 20 years. Naturalis over there is setting the early pace, processing 4 million botany specimens in less than a year. Most institutions start with botany because they tend to be pressed plants, flat, easy to get a nice digital image of. Insects are up next, and entomologists all over the world are positively salivating. Another nice flat object type that are in library and museum institutions all over are books. These are also being digitized in a hurry, primarily by a project called the Biodiversity Heritage Library in partnership with the Internet Archive. And it is hard to quantify this kind of information, but I like pages. That's sort of easy to imagine. And after about 17 years, we've gone past 60 million pages. There's low-hanging fruit with this one too, like starting with the plants. You start with the nicely printed published text, but we all know there's more to recorded science than that, and we are moving on to more challenging material, the assorted vintage unpublished documents of biodiversity science, which I'm sure will inspire lots of fascinating new tools to aid in their interpretation. The second tributary in the data flood is sometimes called citizen science or crowdsourcing. This isn't really a cultural shift. Biodiversity research has a long history being the hobby of the privileged and leisured classes. Nowadays, it can be the hobby of about 2.9 million people with cell phones. It's actually quite easy for a human with a mobile device to create usable biodiversity data, accurately geocoded and time-stamped and vouchered by digital photographs. Identifying what the organism is in the image can be the task of the observer, but there's also a growing online community of professional and amateur experts who can help with this process in a collaborative way. And increasingly these days, machine vision software helps to narrow it down, so it becomes more and more scalable. This particular batch is all observations from here in Leiden in the beginning of February. So you should try it. It's fun. Download the app, go out in the world. If you see me at lunchtime out there in a tree, I'm probably looking for one of those pretty green caterpillars. I'm happy to talk about it. I'm a big fan of this project. The use of iNaturalist has exploded in about the past 8 years. It's very seasonal because people prefer to do this when the weather is nice. And last summer, we surpassed 5 million observations per month. Of course, molecular data is a big contributor also to the data flood, especially for biodiversity science when all you need from DNA is that identify what species it came from. A barcode is cheap, easy, and very sufficient for that task. So I'm sorry about the date axis on that graph. Those accumulation curves from the Barcode of Life data system database are from the fall of 2022 through the summer of 2023. As you can see, they're still accelerating. So all three of these data types are indexed at the Global Biodiversity Information Facility, GBIF, which is the largest aggregator of what we call occurrence data for biodiversity. An occurrence minimally contains a geolocation and a timestamp and some level of taxonomic ID, ideally a species or thereabouts, but that varies quite widely. I love this particular map, although it's out of date, partly because it gives such a glorious representation of our sampling bias. In this picture, purple is the presence of data, and the paler it is, the more data are present. So you can see everything from how resources are distributed on land to the individual tracks of research vessels at sea. You can't get this image anymore. This is back when there were 600 million records available, and that was in 2019. The data have quadrupled since then, so now they summarize with a bubble graph. And even that takes so long to load that I can get a screen grab when it's only halfway rendered. So at least you can still see that there's a map underneath. I find this much harder to read, but you can tell from Antarctica and the Indian Ocean that the sampling bias hasn't changed dramatically in the last 4 years. So 2.6 billion records have drawn our attention to our responsibilities as a data community, and considerable effort has been made to ensure fair data in biodiversity. There are really two kinds of effort primarily that go into making data findable, accessible, interoperable, and reusable. The first kind are policy decisions made at the institutional level regarding licensing, terms of use, and investment in public data services. And a measure of progress in that direction is called the Bouchot Declaration. This is modeled on the Berlin Declaration of 2003, but particularly for biodiversity data, it's a detailed commitment to manage data in order to support open access to biodiversity knowledge. At this point, it has about 100 institutional signatories and a couple hundred individuals signed also. I signed it. So that's the responsibility of the institutions. Having gotten that far, the other part of the effort is at a much more granular level because it requires the individual expertise of staff and workers in general, primarily computer scientists and data managers. And that's a challenge because especially museum data managers span several generations, still actively working, and things are changing so fast that keeping everyone up to date with contemporary methods and best practices is an ongoing challenge. We rely heavily on large, somewhat loose global networks of collaboration and training exchange to support this. Not directly dependent on our home institutions. A big part of this is an organization called the Carpentries, which you may know as software carpentry, data carpentry, or library carpentry. They provide open source online curricular materials and online or in-person training for instructors in data management who then go back to their institutions or their community conferences or any organized event in their region and conduct workshops to bring people up to speed with things like the principles of tidy data, documentation, and reproducibility, and sort of basic accessible tools like R, Python, and OpenRefine. I do OpenRefine lessons myself at the Smithsonian. It's really good fun. People who don't code love OpenRefine. That's why I like it so much. Smaller groups self-organize around particular types of biodiversity data. The Open Traits Network has met a few times adjacent to other larger meetings and mostly collaborate online on documentation for best practice in publishing trait data in particular. There is institutional support for some of this sort of loose collaborative networking. There was a series of meetings, which there have been 3 so far, supported by the German Center for Integrative Biodiversity Research, that enabled a bunch of us data nerds to get together and work out some best practices, tests, and methods. And last time we even had an experienced Wikimedian with us, Daniel Meechan. So by the time that meeting was over, every participant and also the event itself had a Wikidata ID. So that's a start. My community has been what you might call graph aware for about 10 years, ever since our most beloved provocateur Rod Page published this figure, which has haunted us ever since. Obviously, this is just a summary version of the entities in biodiversity data that may have relationships among them. So I'd like to zoom in on the part that I'm most familiar with to give you a little bit more detail. Traits is a term which is used differently a little bit throughout our community. But by and large, we define it pretty broadly. It can include well-defined character states, like the arrangement of leaves on a plant stem. It can also describe categories of ecological role or behavior, which can be either broad or narrow. We use it to document relationships among unrelated taxa. Sorry, among different species. And because it fits the data model so well, some of us even use it for purely human applied attributes like regulatory status. So all of this is data that we try to intermingle for search purposes. Actually, by far the most prized category of trait data are probably measures of size. It's an intuitive sort of thing to ask, and there are a lot of applications for it, notably in global climate modeling. I learned this from the marine modeling community a few years ago. It turns out they like to do things like estimate the total biomass of each component of the plankton community in an ocean basin. So they can then address questions like, how fast does that community deliver carbon to the ocean floor for burial? So size matters. Zooming in a little further, now that you understand what I mean by trait, the project I support, the Encyclopedia of Life, is one of the broader aggregators in biodiversity data. Our mandate is to provide representative information per species, ostensibly for all the species on Earth. We don't exclude prokaryotes and viruses, but we don't love to talk about them because our classification system does rely heavily on taxonomic names, which aren't necessarily adequate for those groups. And we just aren't resourced to provide what we consider a substantial contribution to that area. So we've sort of accepted that those audiences are better served by specialist platforms. But we cover eukaryotes, not badly, I think. The other things you should know about us: all the content that we host has to be freely reusable, either public domain or under a Creative Commons license. We don't create data. We only integrate it from other sources, chiefly specialist online biodiversity databases and the literature. That being the case, detailed provenance is really important for us. It's messy because of the wide variety of different sources and the different ways they are cited. And it takes up quite a bit of data model, not all of which we'll see today. We prize discoverability and portability of data. I actually personally think of EOL not so much as a destination for data users as an index or a navigational aid connecting users with data resources that are not otherwise well exposed. So discoverability is what we strive for. And we try to provide a variety of different access points for different kind of users, because that navigational aid, not a destination thing, means we do get all kinds at EOL. We get these guys. They come for our data services and downloads, primarily bulk use of data. And we also get the general public, the curious educators and learners. And they have a variety of questions, but they've got an uncanny knack for coming up with questions that sound simple, when in fact life is really complicated, even if you're just representing it as representative at the species level. So it is possible to deliver what they need to this wide variety of users, but in order not to mislead anyone, it's important to keep track of a certain level of detail. A typical quantitative trait record on EOL might contain information like this. Not all fields appear in every record. We manage this data in a property graph using a product called Neo4j. So a data record is an entity, And most of these fields are attached to it as direct relationships. So this data record has life stage adult. Adult is a concept that also has an identifier. Um, some values that are, for example, numeric, also literal values, of which we have some, although I haven't shown you because I try to minimize those. Things that you probably won't want to traverse are stored as properties on the data record. Instead, that's a property graph thing. So, for example, this data record has a property sample size with a value 21. These things are conceptually not very different from proximal non-traverse nodes in RDF, and they do take up more space in the data model, but this is what we have. Different kinds of traits have slightly different shapes to their records. For example, a categorical trait record, and these are much more abundant than quantitative ones, will still have the predicate measurement type. It'll just be a different measurement type value. And in this case, because the value is also a concept which you might want to traverse, black could apply to a lot of things, that will be a relationship to an adjacent node. The predicates in that model are primarily provided by a standard called the Darwin core, an extension of the Dublin core specifically for biodiversity data. It has several different sections. This is the section for occurrence data, the stuff that was on that GBIF map. And this provides some of the things that we use like sex and life stage. There are other sections for measurements, for example, that will provide something like sample size. For the objects in the model, we prefer to rely on the oboe foundries wherever possible. We like these because they are mature and stable and the responsibility of someone else. And also because they are richly provided with terms relationships, particularly parent-child relationships. And that's really important for the primary purpose for us of terms management, which is discoverability. So for example, say a user has a fairly narrow data interest and they bring us a query with several facets. The first facet here is size. That's a measurement type facet. This is not uncommon to say, I'm interested in size and not say what kind you want because size data are so in demand and so scarce. Someone is often willing to take whatever kind of size you can give them and they'll look up some estimation formula to get what they actually need. And that's good because size is not consistently reported in the same method. So our terms network for size terms, size is the root and a wide variety of different kinds of measurements descend from there. If you have a particular taxonomic interest, that will rely on our dynamic hierarchy, which is what we call EOL's artisanal internal taxonomic classification. It is a mosaic of a large number of taxonomy data providers. About 2/3 of our 2.4 million taxa are from the Catalog of Life, which is hooked up programmatically, as is NCBI. We get about 50,000 nodes from them. And there are patches that are curated manually. My colleague Katja Schultz handles the problem areas as well as about 9,000 nodes worth of trunk herself, revising by hand every revision. By comparison, geography is very crudely handled in EOL. We have an entirely verbal geographic component to the search. We don't use geolocation in a digital sense. We rely primarily on geonames, and it's not terribly deep, this hierarchy. We go down to nation for most taxa in most areas, and that's just the level at which most of our searchers are satisfied, and you can get good taxonomic coverage if you're about that course. You can access things from several different search directions. For example, you can find Peru in the Western Hemisphere, the Southern Hemisphere, or the Neotropics. Not all of that is provided by geonames. We did have to hook in a few things from other sources. Habitat is a big search facet for us because the data are relatively abundant and they're really easy to organize using the environment ontology. This is natively very well provided with parent-child relationships. Coastal wetland, for example, is both coastal and a wetland. Some areas don't have a single reliable comprehensive terms provider. Don't feel obliged to read this one. It's most important just to sort of appreciate the shape. We struggled with trophic ecology because we kind of had to figure out a lot of it from scratch. This is the napkin drawing that my colleague Katja Schultz made while we were trying to figure it out. And the problems start with the term like trophic yield. You get this sometimes in biodiversity science. A term that's just a big, broad basket. You get everything in there from, this is really almost anything about eating. It covers who you eat, what part of them you eat, how you do it, and a few other unrelated concepts, pretty much all under one term. It's tempting to say we're going to break it down into specialized subgroups, but those will not be terms people recognize. People use trophic yield for search, so we were kind of stuck with it. Anyway, I'm not going to go into all the gory details of the part of this, the particular unintuitive thing that roasted all the brains in my office that day. But if you're curious, do come and ask me about it. We can compare the relative roastability of our brains. Anyway, it's all worth it because we think we have produced discoverability that did not exist before. Google, I think, can't help answer a question like this. Their indexes don't know all the terms relationships that we know. Natural language processing isn't as much help with this as it is with most human questions, partly because the source text where this knowledge originates is not very natural, not uncommon in the sciences. So we think we've contributed the ability to answer this kind of question. This is a bird's eye view of our valiant terms providers. As you can see, the bulk of the values are geographic and habitat. Those are sort of the deepest vocabularies that we have. Wikidata is an important source, particularly for obscure terms. We often find concepts there that are not available anywhere else. And there's a long tail of small numbers of terms that are provided directly by the specialized biodiversity data providers. Some of them manage their own vocabularies. And if they have URIs, we do respect those terms. The best ontology in the world will not help you with discoverability if you only have records for a small fraction of the available species. So what we try to do is push this graph upward before making it any broader. As you can see, there are obvious large gaps, but not all of them are as bad as they look. For example, growth habit only really applies to plants and some sessile marine animals. So 400,000 species is not terrible coverage, but this is an important caveat in the discoverability of data in EOL. I do also comfort myself with the thought that species for which people are searching and that are more likely to be useful to them in their search results are probably the species for which we have trait data available. And if that's not a social media bubble way of looking at the world, I don't know what is. So the searches that we've been talking about can be performed by a human at this interface in our website. It's about what you would expect. You can create a trait search facet using either a measurement type or a value or both. If you choose a term that has children, they will be offered to you. And you can combine a few different trait facets, and you can include a taxonomic facet if you want. You can use this search to ask for a list of species that match your criteria, or to ask for the records underlying them. You can download those records as a CSV file. This can be very wide, this table. It's flat. And a column is provided for any field that appears in any of your records. So, do this if you like flat files, but you can also access the data directly in the graph via a Cypher query. Cypher is a language that has been described to me as a hybrid of SQL and ASCII art. It is arrows connecting bubbles, round bubbles for entities, and rectangular bubbles for relationships. As a non-programmer, I find it really friendly and intuitive, so I think it has potential to appeal to non-programming biologists. We consider an important part of accessibility providing friendly user interfaces for our broad audience. This is another fun thing I hope you will all try. If you come to a taxon page on EOL that has ecological associations data available, it'll give you an interactive display like this, which is a sample of its ecological associates. The taxon of interest in the middle, some of its predator species above, some of the prey species it depends on below, and also to the sides, some potential competitors that rely on the same prey species. You can select any of these species to move it to the middle, summon its entourage, and crawl your way through the trophic web. Also, if you have structured data, it's really easy to construct natural language text, like so much easier than going the other way, right? So we do this for our taxon pages. It's not really important for a species like this that has abundant narrative text available, but it does allow us to provide a sentence or two for nearly all of our 2.4 million taxa. And here's something I just discovered about a week ago while I was working on these slides. I entered the name of this copepod and nothing else into an incognito tab in my browser. And here's Google putting it at the top of the list, despite the fact that there is a richer paragraph available from Wikipedia and giving it this featured snippet treatment. We have a service for this text, but that's not what they did, because this is what it looked like a week ago. I checked again yesterday, and there is now appended at the end an unrelated comment from the source text farther down in the HTML. So I don't know why they did this. Maybe they like uniformity, but this is making our data more discoverable. So much for discoverability. There is one type of reasoning that I thought might interest you that we actually use internally. It's the only thing we do other than search, really, with our graph. It relies entirely on taxonomic relationships, but it affects trait data. So as you know, in life, there are clades where all or almost all the members share a trait. Ctenophores, comb jellies, are pelagic animals. They have habitat pelagic. They swim around in the ocean up in the water column. Great. We create one record, attach it to Ctenophora, and you give it a flag that indicates it is heritable, and paint it down to all the taxa below. But we have to be careful because this is also a ctenophore, highly derived from a group that specializes in crawling around on kelp and things near the ocean floor. Habitat equals benthic. It's okay, we can still use that technique. We just have to put in one more process to subtract the exceptions from this painted node. It's actually a perfectly natural way of recording this information. It goes back more than 100 years on paper. People would write down big group, Ctenophora, free swimming, blanket description, and then make a note of the exceptions. So very traditional. Sometimes the exceptions are just one or two. Some situations are more complicated. Nearly all plants are photosynthetic. Not this one, ghost pipe from my home region of Eastern North America. Really pretty, isn't it? You can tell it's not photosynthetic because it's white. Sorry, none of that near here that you can go see, but if you want to meet a parasitic plant today or tomorrow, this is in bloom right across town, purple toothwort. These are 2 of 660 exceptions to the photosynthesis rule. 660 different branches had to be subtracted. So there on the right is the plants are photosynthetic record in EOL, most of which is a list of exceptions. But this is worth doing because nearly all plants are photosynthetic, and now we're pretty accurate about that. So here's your reminder, download the iNaturalist app. You can go have this pin and go straight to that pretty purple parasite that I met yesterday. Before I leave you, after my own particular adventures, here are some things that I now believe. You do your homework, you choose your tools, you optimize your whole process, and then you wait 10 minutes, check again. Things are changing very fast. You could probably have an easier time doing what you're doing right now if you haven't checked recently for advancements. Do, by all means, leverage what someone else has built. It may not save you time, it may not save you money, but it will introduce you to someone else. And the two of you will have a resource in which you are both interested, and that is worth having. It's slow, but it is advantage you can aggregate. This next one is something maybe not all of you encounter, but I have heard phrases a lot like, oh my gosh, we have too much data. What am I going to do? I can never process it all. You know what? You probably don't have to process it all. Too much data means you have options. You can try different scales and see what difference it makes. You can sample different subsets. You have scope for growth. Don't complain about having too much of something. And this last one has been the most important for me. You created something, you're proud of it, you've invested time and energy, you may be getting attached. Okay. Is it software? Because in that case, it might break your heart. Their lifespan is highly variable. Some of them do stick around, but many of them are useful for a year and then someone has made something else or there's a different route to the same objective. It's okay, it's normal, you should still be proud, be ready to let go and embrace something new. However, if it's a data product and you put thought into it, you prepared it carefully, gave it everything it needed, it probably won't break your heart. And with any luck, it'll go out into the world and thrive and be useful to lots of people without your intervention. And that's important because it'll probably outlive you. Thanks. First talk, important tip.
Okay, so is it okay? Yeah. So I'm just wondering, during the course of my studies of plants and animals, I saw that the taxonomical terms are constantly changing. So it's a huge problem. So how do you cope with this? So it's almost every month there's a change of some clades. So
It's
how do you change?
it's worse than that, because sections that are stable are not necessarily actually stable. There might just be nobody alive working on them. You really just have to use a large grain of salt. And we who are managing the data, it is actually, there's been progress, and it gets easier and easier for data hubs that use automated systems to receive updates from most publishing sources. There are aggregators of taxonomic updates now, and the primary sources, the publishers in the literature, are getting better organized. Hang in there, it will get easier. That's.
But there's no like an international consortium, like the UNESCO-wide thing worldwide.
Yeah, we've been expecting worldwide organization for 30 years, but for the past few years, I think we've realized, no, apparently we're just not going to get that. So it's an organic, dispersed process. I'm sorry.
Good. Thanks a lot.
Thank you very much for the talk. Very interesting. Nice set of data, of structures. So it's very a curious question. Do you have stories of people who take this data and structure to make some discovery, some biological discoveries?
Actual research published?
Yes. Yeah.
So papers are published from the data that we host. I have a link which I will share to, I think it's in my Google Scholar library. Not a large number of papers come from the trait data in EOL that we know of. There is tension between providing open access and being able to track who used data that you did not create, but only disseminated. So normally, if they came to us for the data, it may be mentioned in the acknowledgement section or the methods of a published paper, but it is very hard. The people you need to cite if you take data from EOL are the people who are listed in the attribution section. The primary use cases that we know about, people creating things, are not biological research. They are educational products. There's a tool called Vera, the Virtual Ecological Research Assistant, developed by Ashok Goel at Virginia Tech, which teaches middle school students, the 13 young teenagers, how ecological modeling works, and that is powered by trait data from EOL, so that when they load a gray wolf, they will know how big it gets, how much food it needs, and so on. That's the sort of thing people have done with the data primarily.
Yeah, thanks. Great talk. So in one of your slides, you showed the rapid rise of DNA barcoding and records in bold. And yeah, indeed, that's sort of accelerating exponentially with like BioSQL and so on. And that probably means that in the not too distant future, a lot of taxa are going to be identifiable via molecules and are going to have unique identifiers attached to them, such as these bins or things like that, without having names yet. How are we going to cope with that?
Yeah. We worry about that at EOL a lot. It's actually, we are not unwilling to embrace another system. And I'm pretty confident that one will emerge. EOL doesn't regard itself as the entity that should determine how those systems are integrated, but I'm sure somebody will decide what should be done, and we're happy to follow along. I don't think it should be very difficult. I think, you know, an identifier, a unique string of some kind, is not that different a product from a taxonomic name, apart from the creative freedom that taxonomists used to enjoy. I don't think it'll be that much of a problem. It'll look very different in the databases, I agree, but I think it can be managed.
Thanks.
Thank you very much for your talk. So you primarily use other resources by external parties. Have you had issues with resources going down and not being able to access that data anymore and being able to share that with others?
Yeah, that's a great question, and that's something we worry about a lot. Some of the resources that we have in EOL are now static because the source database is gone. A lot of data sort of migrate from source to source. There are things that we have gotten from more than one place at different eras. And actually, there's a lot of duplication also. That's something that you have to worry about as an aggregator. And a little of that is left as an exercise for the data user. So expect gaps, expect duplication. But yes, we regard part of our role as being the lifeboat for data hubs that go dark. And it can happen to any project, as far as I can tell. Everybody of any project size worries about what will happen when their funding runs out. Our solution at EOL, and I think this is quite common, at least in biodiversity, is ensure the data are portable so some other lifeboat can come along and pick it up.
A second question I had is coming back on the question that the sir next to me just made. So different names changing, etc. You also have that a lot in protein names, in gene names,
Oh, I
in
didn't know that.
I like immensely. 20
sorry.
names, 20 names for one protein, or even more, because every scientist researching something thinks they found a new thing.
Oh, I discovered it,
and
yeah.
they didn't, because somebody else already did, like 20 times 20 years ago. So this is a common issue that we're all having, and I think we can learn from each other as well, like how to deal with these things. So I just think it would be interesting to
That's, yeah, this is news to me that you have the same problem with proteins that we have with species. Absolutely. And it's a whole subspecialty, is handling synonymies. It's not my personal area of expertise, but I know people, and we should absolutely exchange some knowledge.
Yeah, thanks.
So very briefly, thinking about lifeboats, total data size of the EOL?
On EOL?
Mm-hmm.
Oh, you're right. I used to know this, but I lose track. I want to say about 100 million records, but don't quote me on that. It is somewhat more important to consider data coverage across taxa because of the duplication, and just the added value of a 20th habitat record for the polar bear is small. So what I tend to keep track of is how full the tree of life is for any given trait.